What gives axolotls their wild colors and morphs? From snowy leucistics to golden albinos to deep black melanoids, the secret lies in three special skin cells called chromatophores.
3 little cell types equals one big fashion show.
And trust me, it's not tie-dye, even though I'd love to claim it. Hey everyone, welcome back to Maniacal Threadz Exotics. I'm Jay, and today we're cracking the code of Axolotl Morphs. We're going to dive into the science of chromatophores, the pigment cells that act like nature's paintbrush. By the end of this video, you'll be able to look at any axolotl morph and know which pigment cells are behind its unique look.
Basically, you're gonna become an axolotl color detective.
When we say morph in the axolotl world, we're talking about variations in color and pattern caused by differences in pigment cells. Some morphs happen naturally, like wild types. Others come from mutations, like albinos or melanoids. And in captivity, selective breeding has given us new looks like copper or lavender. Scientists also introduced morphs like GFB axolotls, which glow under UV light.
So it's part genetics, part science experiment, and part happy accident.
Let's meet the three types of chromatophores that create axolotl colors. First, we have melanophores. These cells contain melanin, which is the pigment responsible for black and brown tones. The more melanophores an axolotl has, the darker its appearance. Fewer melanophores make the animal lighter in color. For example, wild types are rich in melanophores, giving them that brownish-green base. Melanoids take it a step further. They pack in even more melanophores, but lack the shiny iridophores and the yellow xanthophores, leaving them jet black with a smooth matte finish. On the other end of the spectrum, leucistic axolotls have very few melanophores in their skin, which is why they appear pale pink or white, though their dark eyes remain. In humans, melanin also plays a big role. It decides our skin tone and protects us from sun damage. Axolotls use it in much the same way, but with a far more dramatic flair.
So leucistics are like humans who skip sunscreen their whole lives, pale, but still rocking dark eyes.
Next up are xanthophores. These cells hold carotenoid pigments, the ones that give off yellow, orange, and even red tones. You'll see them shine brightest in albino and copper morphs. Take the golden albino, for example. Because it lacks melanophores, the xanthophores take center stage, giving the animal its glowing golden-orange color. Coppers are another great showcase, where reduced melanin combined with active xanthophores creates those warm, coppery tones. Even some wild types can lean heavy on xanthophores, showing up with bold yellow speckles across their body. Humans, interestingly, don't actually have xanthophores at all. But carotenoids from the food we eat can tint our skin a little, like how babies who eat too much carrot puree sometimes take on a faint orange glow.
So if you wanted to cosplay as a golden albino, just chug carrot juice for a week.
Last but not least, we have iridophores. Unlike the other chromatophores, these don't hold pigment at all. Instead, they use tiny reflective crystals to scatter light. That scattering creates shimmer, pearly tones, and even metallic specks across the skin. In wild types, iridophores show up as sparkling flecks across the body. In leucistic axolotls, they add a soft pearly sheen that overlays the pale skin. And in melanoids, iridophores are missing altogether, which is why their black finish looks so flat and matte. Humans don't have iridophores either, but there's a neat parallel. Our blue and green eyes work the same way. They don't actually contain blue pigment at all. It's just light scattering that creates the color.
It's like diamonds. You're beautiful.
Basically, axolotls don't need glitter glue. They're born with it.
My goal is just to not fall in the fan blades. Oh, it's in everything. I'm gonna do it again. That's a great job on the glitter bomb, because you knew I'd do something stupid, and I did, and now I'm moving. I have to move.
Now, here's where the science comes in. At the University of Kentucky, Dr. Randal Voss and his team at the Ambystoma Genetic Stock Center have spent years studying axolotl genetics. They've mapped out the mutations that create morphs, like the genes responsible for albinism, leucism, and melanoid traits.
At any given time, hundreds of salamanders are being bred at the University of Kentucky.
I direct the Ambystoma Genetic Stock Center, and we maintain the world's largest captive bred salamander population. Ambystoma is the scientific name of the salamander that we maintain. The common name is the Mexican axolotl. Almost on a daily basis, people call us up requesting materials. Hey, do you have some embryos? Do you have some larvae? Do you have some adults? At this time, there are about 150 research labs in the world that use salamanders and depend upon our genetic stock facility, as well as the genomic studies that we're pursuing to help move their research programs forward.
With funding from the National Institutes of Health, from the National Science Foundation, and the Army Research Office, biologist Randal Voss maintains and studies a unique species of salamander that may one day help people with spinal cord and limb injuries.
Mexican axolotls were originally collected from large lakes around present-day Mexico City over 100 years ago. They're amazing because they can regenerate so many of their body parts, their digits, their limbs, parts of their eyes. They can even repair damage to their brains and their spinal cords. Among all vertebrate models, they show the greatest regenerative capacity, and that's why they're an important model. The funding we receive is important because it's allowing us to study the genome of the Mexican axolotl to reveal the underlying basis of regeneration, and importantly, we're sharing this information with other researchers. The axolotl has a very large genome. It's an order of magnitude larger than the human genome. These advances in DNA sequencing technologies that have happened because of the Human Genome Project, essentially every organism's genome will be sequenced. We're taking a very innovative approach. We're using a laser capture microscope that will allow us to isolate single chromosomes one at a time.
Voss's salamanders are interesting to researchers for more reasons than tissue regeneration. They have found a way to postpone growing up.
Typically, amphibians have two phases to their life. In the first phase, they're totally aquatic embryos that develop into tadpoles or larvae. Then, after a period of larval development, they undergo a metamorphosis. They lose those traits or characteristics that were important for living in water, like the external gills and the keeled tail fin that allows them to swim. So at metamorphosis, they lose those traits and they gain traits which allow them to live on land. So axolotls deviate from this pattern. They block metamorphosis and they maintain those juvenile characteristics into the adult phase of life. They truly look like teenagers at the time in which they die.
Voss uses thyroid hormone to turn these perpetually aquatic teenagers into terrestrial adults. This allows him to find the genes responsible for variations in the timing of metamorphosis and genes linked to regeneration after metamorphosis.
Can we identify genes that explain variation in some parameter of regeneration? There does seem to be genes that control the rate of regeneration. It's easy for us to imagine that our eye color can be blue or brown, or a flower can have purple or red color, and those are determined by one gene. But most of the traits that we care about are very complicated in their nature, and how long we live, how much we weigh, or the time at which we metamorphose, or the rate at which we regenerate, is probably determined by hundreds to thousands of genes. And each of those are making some kind of statistical contribution to that overall variation.
In this complex research, support is crucial to providing high-quality salamander resources to biomedical researchers. Shared scientific equipment, like the laser capture microscope he uses to select single chromosomes, purchased with NIH funds, is key to his and other research at the University of Kentucky.
Somebody with very minimal training can go over there and just laser out small numbers of cells, or in this case chromosomes. And so one of the great things about UK is that we've got a number of these kind of common core facilities, and the instrumentation is there, the expertise is there to help you. No single organism provides the best model for every human disease or health concern. NIH recognizes the importance of maintaining a diverse portfolio of animal models, including the Mexican axolotl, given its central role it plays in regenerative biology and medicine. We are proud to be associated with the group of research resource directors to better enable biomedical research.
So if axolotls had a 23andMe DNA kit, Dr. Voss is the guy running the lab results.
Exactly. Thanks to his work, breeders and scientists know which traits can appear in a clutch of eggs and how pigment genes get passed down. Without Voss's research, we wouldn't understand why a pair of wild types might suddenly produce albino or leucistic offspring. So how do axolotl morphs actually happen? It all comes down to which chromatophores are present, or which ones are missing. Wild types carry all three kinds, giving them those earthy tones with gold flecks and a little shimmer. Leucistics have fewer melanophores, which leaves them with pale skin but those striking dark eyes. Albinos, whether golden or white, have no melanophores at all. If their xanthophores are strong, they glow golden orange, and if they're weak, the axolotl appears soft white or pink. Melanoids go the opposite direction, keeping only melanophores. With no shimmer or gold, they're pure dark, with that deep matte finish. Coppers are a special case. Their melanin is mutated, and with active xanthophores, they show off warm reddish-brown tones. Then there are chimeras, one of the rarest finds, where pigment distribution splits in unusual ways, creating patchy blends of multiple morphs. And finally, the famous GFB morph. It's not natural, but thanks to a jellyfish gene, these axolotls glow bright green under ultraviolet light, turning science into something that feels almost magical.
So morphs are basically biology's recipe book. Add a little melanin, subtract some shimmer, sprinkle in yellow, and boom, new axolotl.
Sugar, spice, and everything nice. These were the ingredients chosen to create the perfect little girl. But Professor Utonium accidentally added an extra ingredient to the concoction, chemical X. Thus, the Powerpuff Girls were born! Using their ultra superpowers, Blossom, Bubbles, and Buttercup have dedicated their lives to fighting crime and the forces of evil!
And then there's regeneration. Dr. Elly Tanaka, from her lab in Vienna, proved that when axolotls regrow a limb, they regrow the chromatophores too. That means if a wild type loses a leg, the new one comes back with the same spots, shimmer, and pigment pattern.
Undertaking this amazing feat, which is to grow a bud of tissue after amputating the limb, which then continues to grow and then undergoes patterning to replace the missing part of the limb, and the limb becomes functional. Now, a number of researchers have also used another species of salamander to study this process, and that species is Ambystoma mexicanum, or the Mexican axolotl, that was collected over 100 years ago in Lake Xochimilco. This is a wild-type axolotl. You can see its beautiful skin pigments. In nature, there are also other natural mutants, such as this one called the white mutant, or DD, which lacks skin pigments. And this is helpful for us because it allows us to see the cells in the regenerating tissue as they're undergoing the process of regeneration. These salamanders can lay up to 500 eggs at a time. And these animals become fully active and functional quite early in their lives. This shows you a young axolotl, and at this stage the animal has a forelimb, although it doesn't have a hind limb yet, and we're able to study regeneration in these size animals. This helps us to put the animal under the microscope and watch the process of regeneration. Let's go over the parts of the limb that we're going to be talking about today. So as you know, in our limbs, we have an upper part of the limb, we have the lower arm, and then we have the hand part of the limb. In addition to the 3 parts of the limb, we have the different tissues that make up the limb. We have the skin marked here in blue, muscle fibers marked here in red, and then Schwann cells and nerve cells marked here in green, and bone marked here in black. Knitting many of these tissues together is also a cell type called the fibroblast, which is diffusely around many, many different tissues. Now let's look at the structure of the blastema. This shows you a time course of the regeneration process again, the formation of the blastema as it undergoes growth and patterning to regenerate the limb. If we take one of these young axolotls undergoing regeneration and put it under a microscope, we see this. You can see this is the upper arm bone, and these are the lower arm bones. And this amorphous structure here is the blastema. If we now take a thin section through this tissue and stain it with some dyes, we can see the cells inside. So these are the lower arm bones, and this is the blastema. And you can see it's covered by a piece of skin called the epidermis. So this is where all the action is happening. These are the cells that are going to regenerate the new part of the limb. How do they do it? Well, today we'll address 2 fundamental questions that we need to understand the process of limb regeneration. First, which cells from the adult part of the limb are recruited to form this blastema? Secondly, how does the limb regenerate only the missing part? So let's start with this first question here. Now, researchers over many years have been examining this question by different methods, and I'll illustrate for you 2 of these experiments. For example, Trygve Steen found a way to label limb cells of the salamander by providing them with radioactive nucleotides that incorporated into the DNA of the cells of this animal. He then took a small bone part of of a limb from one of these labeled animals, cleaned it as well as possible, and then placed it into the blastema of an unlabeled animal. When he then followed regeneration, he saw that the labeled cells were taking part in regeneration, but produced only bone cells. So he concluded from these experiments that bone-forming cells only form bone during regeneration. In contrast to these experiments, an experiment conducted by Hugh Wallace and Malcolm Maiden suggested that Schwann cells might be able to produce many different cell types during regeneration. Interestingly, in order to inhibit regeneration, they treated the limb with X-rays. This prevents the process of cell proliferation so that when the limb is amputated, it is unable to regenerate. In order to rescue regeneration, these researchers then transplanted a piece of nerve, a normal piece of nerve, from the unirradiated animal into this irradiated structure. When they waited over time, then an entire limb regenerated. So this raised the possibility that Schwann cells might be able to lose their specialized property in a process called dedifferentiation and produce many different cell types. But of course, it was also possible that their grafted tissue might have contained some unknown cell type that also could have contributed.
That's like losing a shoe and having the universe send you a new one in the exact color and size, custom ordered by nature.
Yes? Can I help you, sir?
Can this, please?
Certainly, sir. You are, sir, size 9.
No, but you want the...
It's more than cool. Studying how chromatophores regenerate gives scientists clues about how human skin repairs itself, and why sometimes pigment cells misfire, leading to conditions like vitiligo or even melanoma. If you're enjoying this mix of science and axolotl magic, hit that like button and subscribe. It helps us keep building this colorful community.
Trust me, it's easier than keeping a tank clean.
Pigment cells aren't just about looks. They're a window into bigger biology. Studying morphs and chromatophores helps us answer questions about genetics, cell development, and healing. It shows us how animals adapt, how mutations work, and even how we might one day repair or regenerate human tissues more effectively.
Axolotls aren't just pets, they're living blueprints for science.
So whether you're staring at a sparkling wild type, a pale leucistic, or a jet black melanoid, remember, you're looking at the work of chromatophores, genetics, and decades of research. Scientists like Dr. Randal Voss and Dr. Elly Tanaka are helping us connect axolotl biology to human biology in ways we're only beginning to understand.
Axolotls, cooler than crayons, shinier than glitter, and way more stylish than humans.
No, stop right there. You're not a crayon. No.
Thanks for hanging out with us here at Maniacal Threadz Exotics. Until next time, stay curious, stay colorful, and keep exploring.
If you're interested in adopting one of our babies, drop a line in the comments below or email us at maniacalthreadz@gmail.com. I will drop the info in the description below. Until next time, water dragons, stay kind, stay true, be quirky, and spin into the colors of the universe.